Here's our quick roadmap for today. We'll start with the mysteries of the brain, unpack the resolution trade-off, explore direct measures like EEG and MEG, shift over to blood flow trackers, and then finally look at how to choose the right tool for the job. Starting us off with Section 1, Mysteries of the Brain and the Subjective Experience. To really get these tools, we first kind of have to understand the sheer scale of the challenge. We're trying to translate entirely subjective, personal human experiences into objective, hard, measurable data. Take the profound phenomenology of a near-death experience. People report phosphenes, those sudden flashes of light. They hear ringing in their ears. They experience crazy out-of-body perspectives, a sensation of traveling down a dark tunnel and eventually entering a bright light. Mapping these fleeting, deeply personal events inside a dying or traumatized brain introduces literally the ultimate scientific challenge. How on earth do you map a vision? How do you record an out-of-body sensation? That brings us to Section 2, the resolution trade-off, where versus when.
So brain imaging is a delicate balancing act. There's no perfect magic camera that does it all. Let's break down this trade-off. The where is what we call spatial resolution. This is the precision of our measurement in space. Basically, high spatial resolution means we can pinpoint the exact millimeter of active tissue within the brain's dense physical structure. If you want to know which tiny little fold of the cortex is lighting up when you look at a face, you absolutely need great spatial resolution. And then on the flip side, we have our when. That's temporal resolution. This measures the exact fraction of a second a cognitive event occurs. The brain moves incredibly fast. Thoughts happen in milliseconds. If you want to track the exact sequence of a visual reaction followed by semantic understanding, you need high temporal resolution.
Let's look at Section 3, Direct Measures. These are our when tools. Let's start with the tools that prioritize the when. These are our direct measures. They act incredibly fast because they pick up the actual electrical and magnetic signals generated by neural activity in real time. Often, studies will use an EEG, an electroencephalogram, which uses 64 or more electrodes placed really precisely all across the scalp. These electrodes measure changes in electrical potentials. But because the electrodes are sitting outside the skull, the signal is kind of blurred. Think of it like standing outside a massive sports stadium. You can't hear individual conversations in the stands, but when someone scores a goal, you instantly hear the roar of the crowd. EEG is exactly like that. It gives us brilliant millisecond by millisecond timing, so fantastic temporal resolution, but pretty poor spatial resolution. Now, what's really interesting about this next direct measure, MEG, is that it takes a slightly different approach. Magnetoencephalography captures the incredibly weak magnetic fields generated by those primary electrical currents in the brain. Because magnetic fields just don't get as distorted by the skull as electrical fields do, MEG actually gives us better spatial accuracy than EEG. You can localize activity much more easily. But there's a huge catch here. These magnetic fields are so unbelievably weak that the engine of a bus, a whole kilometer away, produces a stronger magnetic field than your brain. Crazy, right? So to measure it, you need an astronomically expensive, heavily shielded room just to block out the magnetic noise of the outside world.
Moving on to Section 4, Tracking Blood Flow, the where tool.
Okay, so if EEG and MEG are super fast when tools, what about the where? Well, actively pitting direct electrical measurements against indirect blood flow measurements makes the ultimate resolution trade-off crystal clear. These indirect methods trade that down to the millisecond speed for incredibly detailed structural and functional maps, and they do it by tracking something much slower than electricity, blood flow. Electrical signals are fast, but really hard to pinpoint through the thick skull. Blood flow, on the other hand, takes a couple seconds to catch up to a thought, but we can map exactly where that blood is going with stunning millimeter precision.
So, the crucial point is how fMRI actually accomplishes this. Functional magnetic resonance imaging relies on massive magnets, sometimes weighing up to 11 tons. So, yeah, definitely remember to leave your keys and credit cards outside for sure. Here's how it works. When a brain area becomes active, it consumes oxygen. Blood then rushes to that specific area to replenish it, which changes the ratio of oxygenated to deoxygenated blood. And it turns out, deoxygenated blood actually has magnetic properties that distort the fMRI's magnetic field. This is called the bold signal. It's almost like a satellite tracking traffic jams to figure out where a stadium is located. It gives us this beautiful 3D map of the active brain, but again, it has a delay of about 2 seconds.
Next up, Section 5,
positron emission tomography, or PET, tracking isotopes.
So, before fMRI became the absolute gold standard for spatial mapping, researchers heavily relied on PET scans, which is an older, semi-invasive technique. To track blood flow this way, researchers actually have to inject a radioactive tracer directly into the subject's bloodstream. And the physics behind this are brilliantly fascinating. The radioactive isotope travels to the most active, blood-hungry parts of the brain. As the isotope decays, it generates positrons, which then annihilate with electrons to emit gamma rays. The PET scanner is essentially just a giant ring of radiation detectors that catch those gamma rays. The downside here? It takes about a full 30 to 60 seconds of activity to build a clear picture, so its temporal resolution is incredibly slow, up to a full minute. Because of this, and the fact that it's somewhat invasive, it's rarely used in modern cognitive research compared to fMRI, though it still has really great clinical uses.
Finally, Section 6. Choosing the right tool. The investigator's cheat sheet. Let's bring it all together and see how this builds. As a focused learner stepping into the vast world of neuroscience, the main thing to take away is that there is no single perfect brain scanner. It depends entirely on your specific cognitive question. Are you looking for the exact speed of a reaction? Or are you looking for the specific neural network processing an emotion? That brings us to our investigator's cheat sheet. If you want high temporal resolution to see the time order of brain events, you're going to use EEG or MEG. If you want high spatial resolution to see exactly which networks are performing a task, you're going to use fMRI or PET. Solidifying these acronyms and their unique trade-offs is absolutely crucial for understanding how scientists map the mind. Which really brings us full circle back to where we started. When someone experiences a sudden flash of light, an out-of-body sensation, or profound mystical event at the very edge of life, it's these exact non-invasive tools that stand ready to decode it. As technology continues to improve, slowly minimizing the trade-offs between space and time, we have to ask, which tool will eventually unlock the ultimate mysteries of consciousness itself? Thank you so much for joining me on this explainer. Keep asking those big questions and keep exploring. Catch you next time.